putting together this nice workshop. And this talk will be about evolution. Okay, my point of view is that the name of the workshop is Physics and Biology of Proteins, and really proteins are interdisciplinary objects. You have to look at them both from the point of view of physics and under the point of view of evolution to understand them. And of course, the interesting question that I will address, that interests me, is what are the properties of proteins that are naturally selected? Okay, this is consequences both for the physics, because these protein properties, of course, will be physically important properties, and from the point of view of evolution. If some of these properties have to be trade-off, there is a kind of synergy between them, negative or positive, then this will influence the way in which proteins evolve. Okay, so of course, to do that, we need very, very simple models of proteins. Okay, we have to simplify to the string to be able to understand both physically and evolutionarily. So I will what I will talk will be based on contact interaction models, in which you simplify the of the protein just with the contact matrix. It's unfortunate that Hugh Song-Chan did not give the previous talk because, but I'm sure that you know a lot of this very simple representation of proteins, in which you just ask yourself whether two residues are in contact in a structure or not. And this representation is endowed with a distance measure based on the contact overlap, so how many contacts are in common between two structures. And it's useful to generate alternative structures, for instance, protein-like structure, for instance, from the protein data bank through the procedure which is called threading, that consists in aligning a protein sequence to protein structures. And you have very simple free energy, contact free energy, where the effect of the solvent is averaged out. That depends on these parameters, so the contact interaction between amino acids of types, for instance, leucine and disoleucine, it has 210 parameters. And if you study lattice models, you realize that you can have sequences that are well designed, okay, that have these kind of properties, that all the structures with low energy are very similar to the ground state structure that you can call the native structure of this model. Okay, so these models will be in particular thermodynamically stable, because all the parameters have to be thought as Boltzmann average over the degrees of freedom that we do not consider, and in particular over the degrees of freedom of the solvent. So if the temperature of the pH changes, the contact interaction parameters will change, but the ground state will remain stable. And also they will be stable against mutations. And since energy and escape is well correlated, you can indeed demonstrate with simulations that the ground state is easy to reach kinetically. While in this situation, which is a random sequence, then you see that you have a structure that has a very reduced energy gap with respect to the ground state, but it has a overlap very, very small. So it's completely different structure. It's a compact but misfolded conformation that will act as a kinetic trap during kinetics, and will become the ground state when the thermodynamic conditions change and upon mutation. So this kind of model does not represent protein, because it's not stable against misfolding, and you can assess it through the normalized energy gap, for instance, variation of what has been proposed by Shaknovich a long time ago. So you can use this picture to determine parameters that work well. you have in the PDB you have lots of native structures and their sequences, and you want to optimize the Boltzmann average of the overlap between the native and alternative structures generated, for instance, by threading. And if you do that, then you can find parameters such that for most sequence structure pairs in the PDB, the ground state, so the native structure has minimal energy, and the energy landscape is well correlated, so the energy gap is large. So this model is stable against misfolding. And this model is very simple to interpret because contact interactions basically boil down to hydrophobicity. Okay, if you make a spectral decomposition of the contact interaction matrix, it was realized a long time ago that the main eigenvector of the contact interaction matrix is related to hydrophobicity. So essentially this model, what does it do? If you optimize the sequence under this model, what you will do is to place hydrophobic residues in positions where there are many contacts in the interior of the protein, and hydrophilic and polar residues in exposed the positions where there are few contacts. And this very simple rule is actually useful because it can rationalize a lot of things of protein stability and evolution. And well, these parameters also give some reduced but statistically significant predictive power in predicting the effect of mutations or predicting the delta G of simple proteins that fold with a two-state mechanism. So we want to apply this model to detect which are the features of protein stability that are selected. And of course, we know that there are two kinds of problems that folded protein must solve. One is stability with respect to the unfolded structures, and the other is stability against wrongly folded, misfolded conformations. So people have given different names to these two kinds of stability. So positive design are the features of protein sequences that enforce stability with respect to the unfolded state. So it's you want to make the native structure more stable. But you have also to consider negative design. Okay, because if you make the native structure very stable, you can also stabilize the other compact but misfolded conformations, wrongly folded structures that would be a problem. So the negative design are the characteristics of protein sequences that destabilize compact conformations that can be easily formed. Oops, sorry, what does it happen? Okay, and to study it, we need a model of the misfolded ensemble. So it's a long time that this model exists. It's just based on the contact interaction model, and you can compute its free energy. You can realize actually that you can compute it this free energy through cumulant expansion of the Boltzmann factor, and just the first two models will be two moments of the expansion, the random energy model by Derrida, but you also need, we've shown that you also need the third moment, but you can compute these average values and fluctuations of the energy through the average values and the fluctuations over the native contacts over the ensemble of alternative contacts. Okay, and you have access to the statistical properties of the contact matrices, but just by looking at the contact matrices that are realized in the protein data bank. So, okay, from this model, the first thing that you can see is that selection for positive design is weak for long proteins. Long proteins have the fraction of contacts per residue, number of contacts divided by number of residues, grows with the length of the protein because the surface to volume ratio goes down. So you have more residues in the interior, you have more contacts, and then it's sufficient a small stabilization energy sufficient to compensate the conformational entropy loss due to folding. So you expect that the contacts are less, the native contacts are less, must be less optimized with respect to the random non-native contacts that act as a random background. And this is what we observe from real protein in the PDB, that indeed the longer is the chain, the less optimized are these contacts with these native contacts, the energy of the native contacts, which is consistent with the fitness stability model that tells you, okay, that associates the fitness to the fraction of the protein that is folded in a native condition. When this fraction is close to 1, then an improvement in stability will be almost neutral because it would not modify substantially the fitness. Then evolution cannot fix this, fix larger stability. Okay, so this was noted by Taberna and Goldstein a long time ago, and this is completely consistent with this result. So the selection for positive design is weak for long proteins, but the contrary happens for selection for negative design. Okay, we can see that the freezing temperature of the model, this is the freezing temperature, increases with that you can compute this kind of random energy model, increases with chain length. So longer proteins have a freezing temperature of the enfolded ensemble which is higher, so that room it's more likely that the misfolded ensemble is almost frozen at room temperature. And of course, as I told you, the main component, it is computed. Yeah, this is the frozen temperature of the model. Of course, this is not real experimental data, but it's quite clear that it increases with the length, while the free energy of the misfolded ensemble becomes more negative, so it becomes stabilized the more hydrophobic is the protein sequence. So because of this, we expect that more hydrophobic sequences and longer sequences will be subject to more selection for negative design. And how does the negative design act? Well, the contacts that are close along the sequence, of course, they have to pay a smaller entropic cost to form. And then these contacts with small I minus j that will be more frequent must be destabilized by, we expected that they are destabilized by negative design. This is actually what happens. This is, okay, so these are the contacts that have highest energy and that are less stable, and these are non-native contacts that are close along the sequence, and also native contacts that are close along the sequence that are here, that are also represented here, have high energy, are less stabilized because of negative design. So this is a prediction of the model that is fulfilled. And also we can define the score, the contact frequency score, and we can also define a score for contact correlations, because there is also the second moment of the contact energies that acts on contact correlation. So if this score is positive, it means that it's better for a negative design. And you can see that this, as we expect, this score is higher the more negative is the average interaction energy of the protein sequence. So these are real proteins from the protein data bank, and this is a Z-score of the contact frequency score or the contact correlation score with respect to reshuffled sequences. Okay, certainly the same amino acids, but reshuffle the position of the amino acids in such a way to have a random background, and you see that all these scores are positive, which means that the negative design is significant, and it's more significant for sequences that are more hydrophobic, so the interaction energy is more negative, and for sequences that are longer, as it is expected from the model. So the expectation is that this kind of more hydrophobic and longer sequences will feel more selective pressure for negative design, and this is what you observe. Okay, I think this I already showed. And but superimposed to this, there is an interesting observation. So a long time ago, Stephen Plotkin proposed a model that says that the folding rate of proteins is enhanced if short-range contacts are more attractive. And this goes exactly against negative design. For negative design, we want that short-range contacts must be repulsive, must have a high energy. So indeed, what we observe is that these very short-range contacts are repulsive, as predicted by negative design, but for constants on the range of 8 to 10 amino acid distance along the sequence, we see that indeed there is an excess interaction energy with respect to what the model of negative design would predict, exactly in this range. So for us, this is a kind of a confirmation of Plotkin's model that probably there is some selection for fast folding, and of course you do not expect this. Just a small amount, yeah. Yeah, it's a very small amount, but what convinced us is that this amount increases with the absolute contact order. So the larger the absolute contact order, the slower is the folding of the proteins, and we expect also that selection for accelerating the folding will be larger for proteins with large absolute contact order, and this is actually what we observe in this. So we think that the interpretation is correct. This excess in interaction energy is indeed due to the selective force to accelerate the folding, which does not exist in all proteins, but only in some of those, and in particular in some of those that have large contact order. So, okay, positive and negative design, how are they related? Well, as you can expect, they are inversely related through hydrophobicity. Call it interactivity because this is not any experimental hydrophobicity scale. It's the hydrophobicity scale which is given by the main eigenvector of the contact interaction matrix. So the interactivity, but it's very strongly correlated with experimental hydrophobicity scales, and there are many of those. So the more hydrophobic is a protein, the more stable is. Each point represents a proteome of an organism. So the 20 orthologous proteins in the same organism, and they are characterized by different hydrophobicities. Oops. And they are so the more hydrophobic is the proteome of the organism, the more its proteins are predicted to be stable against unfolding, but they are predicted to be less stable against misfolding. So, which is somehow consistent with observations, right? Proteins that do not have a hydrophobic core are disordered, and proteins that have too many hydrophobic residues, they tend to have intermediate states that act as kinetic traps. And we think that this is so what the force that decides whether a protein is more stable against unfolding or against misfolding is an evolutionary force, is the mutation bias. Okay. In bacterial genomes, there is a broad distribution of guanine cytosine content, and very more broadly the third codon position, which is here. You have a variation of the GC content first position, which almost spans all the possible values. But the GC content of first and second position is strongly correlated with the third position. And since they respond to different evolutionary forces, because the change from G to A at third position is mostly synonymous, and the position from change from C to T in third position is almost synonymous. So the third position responds very little to changes in the protein sequence, whereas the first and second position responds very strongly to that. So this correlation, in our view, is not due to selection, but is due to the mutation. Okay, the common force that acts both in the third position and the first and second position is mutation, and this is there are several interpretations of the variation of the GC content bacterial genomes, but the main interpretation is actually due to mutation and to the fact that the mutation process depends on the correction, on the replication and repair genes of each organism. Okay, and the GC content is strongly related with the hydrophobicity, because thymine, uracil, in second codon position is a mark of hydrophobic residues. So there is a strong correlation between the GC content and first, second position and hydrophobicity. So we computing, predicting these stabilities with respect to misfolding that we measured through the normalized energy gap of several bacteria, we saw that there is a correlation with genome size, and the triangles represent intracellular bacteria that are characterized by reduced effective population size, because they are symbiotic bacteria that cannot live outside of their host. They are transmitted from one generation to the other, and this transmission is like a very strong bottlenecks that reduce the effective population size. And they are characterized by strong AT bias. Their genomes is very rich in thymine, so we predicted the proteins are very hydrophobic, and then we expected that they have reduced stability against misfolding, which is what is observed here. Reduced stability with respect to misfolding. So to see whether all this is consistent with the model, we simulated the fitness stability model, in which the fitness is essentially the fraction of the protein which is folded, and delta G is the folding free energy that we can compute with our simple model of protein energetics. And then from population genetics, it is known which is the probability to accept a mutation when there is a change of fitness. Okay, it's such that for large population size, you never accept a deleterious mutation, but if the population size is small, then this is more tolerant to accept deleterious mutations. Okay, we simulated, and this is the stability with respect to misfolding. This is the stability with respect to unfolding. And do we see that they are indeed inversely related in the simulations as they are in bacterial proteomes? And from one simulation to the other, what we change is just the GC content of the mutational process. Okay, so if the GC content is large, then the proteins will be more stable with respect to unfolding, but less stable to respect to if it's large, it will be more stable to respect to unfolding, but less stable to respect to misfolding, and the other way around. So this trade-off between stability with respect to unfolding and misfolding also happens in the model, and the consequence of this trade-off is that there is a maximum in the fitness that depends on the mutational process. Okay. For a given population size, we find the maximum of the fitness given GC content, and at least in the model that we simulated, it happens that populations that are small have a preferred GC bias that is small, so they prefer to have more adenine and thymine in their genomes, exactly as it happens for intracellular bacteria. that also have a mutational process that is biased towards adenine and thymine. So, but we found an experimental demonstration that the mutation bias is indeed under selective control in experiments of the evolution of foot and mouth disease virus subject to mutagenic pressure due to fluorouracil. Okay, when you replicate these viruses in the presence of the mutagenic absent fluorouracil, then there is a loss of fitness. But suddenly, the group of Esteban Domingo found that a mutant, a mutant strain that has a mutant polymerase, and the effect of the polymerase was not to make replication more accurate, so to reduce the mutation rate, but to change the mutation bias. Okay, we found that changed the mutation bias, in particular in presence of fluorouracil, that the proteins produced by replication in the presence of fluorouracil lose a lot of hydrophobic residues, and the mutant polymerase reduced this loss of hydrophobic residues. And we predicted that the proteins produced were less unstable. So mutation bias influences the balance between unfolding and misfolding, and the stability model predicts that there is an optimal mutation bias that corresponds to optimal hydrophobicity and depends on population size. Proteins of intracellular bacteria less stable with respect to misfolding, both due to the bias towards adenine and thymine and due to the reduced effective population size. And these bacteria overexpress chaperones, which is a very costly way to take care of protein folding problems. Okay. So, and that has been observed in experiments that chaperone overexpression is also able to rescue fitness loss due to experimental population bottlenecks when the very small amount of a population is passed from a plate to another, and this reduces the effective population and increases the stochastic effects of replication. Then there is a fitness loss, and this is mostly due to the loss of stability of proteins, because it can be rescued through chaperones. So this suggests that mutation and selection are not independent. Mutation can limit the results that selection can achieve, and selection can act on replication genes and error correction genes, so that the mutation is under genetic control. Okay, so the last point to make, how to introduce these results in models of molecular evolution. Okay, it was observed a long time ago that proteins that are considered as the same fold, or that are very similar topology, have sequences that are very small sequence identity, as small as for random sequences. But we ask ourselves, what are the statistical properties? Because it's important for modeling molecular evolution and also for bioinformatics applications such as fold recognition and protein alignments. So we can use once again this Moran's model of acceptance of a mutation given its fitness change, and it was realized in 2004 by Sella and Hirsch that indeed a model in which there is only one mutant at each time, and you have to decide whether the mutant gets fixed on the population and it's eliminated by natural selection, is just a Monte Carlo process in sequence space that goes to a stationary distribution, which is completely analogous to a Boltzmann distribution. Okay, where the inverse of population size plays the role of temperature, so smaller populations are more tolerant to the deleterious mutations, and the log of fitness plays the role minus the energy. So large populations attain larger fitness, And the stationarity, there is a balance between mutation and fitness, so that positive mutations, substitutions that increase fitness are as abundant as substitutions that decrease it, which is a bit anti-intuitive for. So, okay, with this Boltzmann model, you can actually model the distribution of amino acids just by making the hypothesis that it is the distribution in sequence space that has minimal Kullback-Leibler divergence, which is analogous to the entropy, from the mutational distribution, which is constraint on the average fitness. And okay, this is, of course, just wording, but you can analytically solve this model in the independent size approximation, where you say that the distribution of amino acids is just the product of the distributions at every position, which is the standard assumption of molecular evolution in bioinformatics. All the computations that are based, molecular evolution computations that are based on likelihood computations cannot be performed without this simplifying, without this assumption. And this is also what is assumed in hidden Markov models, where you have profiles at every position of the protein. So although under the point of view of physics, it's a very simple approximation, mean field approximation, but it's still interesting for biology. So fitness is modeled from fold instability, and there is no free parameters because we have our model. Of course, we don't believe it blindly, but it's a reasonable model. And we only have to determine selection parameter lambda, the Lagrange multiplier, and the background distribution, which is the same for all sites. Okay. So we can determine these mutational and selection parameters by maximum likelihood, by imposing that the sequence that we observe in the PDB has maximum likelihood with respect to the model. And this is the likelihood as a function of lambda, so you can see that there is a well-defined maximum that helps you to determine the selection parameters. And you can see that the likelihood is it's much higher if you consider stability with respect to misfolding. So you also consider the model of the misfolded state, which is the black curve, than if you consider only selection with respect to unfolding. So you don't consider the misfolded state, but only the unfolded state. And in particular, if you neglect the misfolded state, you have these rare curves of hydrophobicity. So you overestimate a lot hydrophobicity. Okay, what limits hydrophobicity is this need to destabilize misfolded conformations. So in particular, here you can see the same over the previous graph was just one protein, and this is average results for 280 proteins, and you can see that if you consider only the mutational process, you can improve the likelihood in considering selection with respect to unfolding, and you can improve it more considering more moments in the of the misfolded ensemble. And if you consider the full model with the selection with respect to misfolding, you produce a model with average free energy will be on the average negative. So most of the models that you predict in this way correspond to proteins that are on the average stable, whereas if you only consider selection with respect to unfolding, or you can only consider the background mutational distributions, of course you lose stability completely. And with respect to hydrophobicity, if you consider only selection with respect to unfolding, you overestimate the hydrophobicity a lot. Each point now is a protein, and it's necessary to consider stability with respect to misfolding. And also this model has the property that if you parameterize the mutational process with a GC bias, then there is optimum fitness that is achieved at the intermediate GC biases in the simulations. Okay, so this model is very simple to rationalize because hydrophobicity is its main component. So here it's just one protein, and we represent every site by the number of contacts, and you see that the average over the model of the hydrophobicity increases linearly with the number of contacts, as you would expect. So in the sites in the interior which are have many contacts tend to be more hydrophobic. But the variability of each site depends on the background mutational process. So when the hydrophobicities of the site is equal to the average hydrophobicity of the background mutational process, you have a maximum variability. You have the least constraint, and you have the largest sequence entropy of this model. And this is a feature of the model that we tested. And okay, so replications, you can see that the distribution of amino acids that you obtain is improves with respect to not to using these site-specific models for all protein families, and also if you perform phylogenetic inference, the likelihood of the model improves. So the Akaike information criteria, so the number of parameters minus the likelihood improves in almost all cases, except for very low sequence identity with respect to using the common empirical molecular evolution models. And we can also use it for ancestral sequence reconstruction. But okay, to test this model, we looked at the substitution rates. It's useful to have a predictive model of substitution rates because this could be a kind of functionally neutral model for detecting high substitution rates due to positive selection. Okay, in this model we only put the positive selection that is imposed by stability, which is a not interesting selection. Of course, the protein does not make anything new, just preserves its stability. And the rate of substitutions at different protein sites and also different proteins has been used as an indicator of positive selection. So that something interesting is happening for the protein and is selected by natural selection. So it has been observed a long time ago that the substitution rate at the position of the protein correlates with the relative solvent accessibility, and indeed it has been shown by Julian Echave and others that this correlation is mainly due to the number of contacts. Positions that have many contacts that are in the interior of the proteins tend to evolve more slowly. And this observation has been interpreted in the sense that exposed sites are less subject to structural constraints, and two models have been proposed to explain that. One is the stability model, that is essentially the same model that I described to you, where what is really important for natural selection is the stability of the protein. And the other model is the structural based model proposed by Julian Echave, in which a mutant, the structure of the mutant changes, and you can predict the extent of this change using elastic network models. And this is really what matters to natural selection, that you want to preserve the functional structure of the protein. So to distinguish between these models, we looked at the substitution rates. Site-specific substitution rates can be computed through these molecular evolution models, in which you have these site-specific amino acid frequencies that we estimate through our model, and then you have exchangeability matrices that are also represented at each position, that represent both the mutational process and the fixation probability. And if you do this for real, real data obtained from multiple sequence alignments, you see that the substitution rate is positively correlated with sequence entropy, as you would expect, and that the substitution rate is smaller for positions that have more contacts. Okay, the more contacts, the more structural constraints, and the slower is the evolution. But in the model, in the stability model, this does not happen, and because we have this maximum of the sequence entropy for intermediate number of contacts, there is also a maximum of the substitution rates for intermediate number of contacts, which is not consistent with observations. And the crucial feature for having this is selection on misfolding. If we eliminate from the model selection on misfolding, then we see that more positions with fewer contacts evolve faster, because we are not taking into account negative design. Is this really negative design what prevents that these highly exposed positions can be too hydrophobic? So we interpret this as that there is a conflict between the data and the model, and we interpret this as a port to the structural based model, where selection acts on the native structure. As you can see, okay, I will skip all of this, but using elastic network models, you can see that there are dynamical correlations that you can compute, and that these dynamical correlations are strong in positions that are in the binding sites of proteins, strong in the active sites. There are deformation couplings that are the basis of the allosteric effect, and we found that these couplings are strong. These deformation couplings are strong between the allosteric regulator site and the catalytic site. And okay, so of course, a more realistic model of protein evolution must take into account also the effect of the protein structure on protein dynamics, and of protein dynamics on function. Okay, I just terminate acknowledging many collaborators over many years, and thank you very much for your attention.
Okay, we thank the speaker. And are there any questions?